Kami sorry kan masih kelas kita lah.
Ya, layankan suara Amir di belakang. Layankan suara Amir di belakang. Nafi alami sekali.
Hai Aziz Zulkifli.
Ya.
Hai Eli Ariana.
Subhanallah. Dua tu je tak guna betul. Right. Anyway, live stream has started. So let's continue with the presentation. Semalam kita dah I think yesterday we finished. I think yesterday we finished on how males inherit hemophilia and how females can become carrier for the condition. Well, semalam was a bit rushed. So do you want me to explain further or we can skip this? Hello?
Dua-dua pun boleh, Aib.
Dua-dua pun boleh, eh? Right, so let's look back on the question, but this time slower. So the question C. Apa dia?
Lukis Punnett Square.
Lukis Punnett Square. Boleh. Tak ada.
Cerita semalam.
Say, give me time to masuk Zoom. But anyway, again, as I've stated yesterday, when discussing the question, when the question says explain rather than outline, the explanation for the thing has to be longer in the sense that you have to explain each of the process in great, each of the process in detail. All right, you have to explain each of the process in great detail. Therefore, in this particular case, we have to explain how hemophilia is inherited in both male and female. But the question specifically asks for the question specifically asks for how males can inherit hemophilia and how females can become carrier for the condition itself. So anyway, let's try drawing a Punnett grid for hemophilia. If so, allow me to share. I'm gonna share the screen for a while. Kita slow dah sebab ada 2 period. So jom kita try. Alright. Alright, can you guys see the screen?
Can.
Alright, so let's begin. Okay, so as usual, before you start drawing a Punnett grid, make sure you define each of the alleles that we're gonna use. So the alleles, we know that hemophilia is a sex-linked gene. Just annotate it somewhere. It's a sex-linked gene that is found on the X chromosome. So by this, it is found on the X chromosome. So by writing this down, you guys would have gotten some marks already because the command was explained. So that's the first thing. The second one, you have to say that male has an XY chromosome. Male is XY and female will have an XX chromosome. That's the second part, and you guys should have gotten a mark just like that. And the third part, explain that because male have only one X chromosome, therefore, as long as they have the allele for hemophilia, the recessive trait will be expressed. So males cannot become carriers, but rather male must become male will have the condition if the allele is located on the X chromosome of the male. As for female, you guys have to explain that female can either become carrier if they have one X with hemophilia with, I'm just gonna annotate it as HF, hemophilia, and one X with no HF. Female will become the carrier. And the fifth one, female will get, okay, one, you guys might think that female, we should explain also how female can get hemophilia. Tapi if you notice in the question, dia tak minta pun macam mana female boleh menghidap hemophilia. So we can willfully ignore that if you want to, because it's not in the mark scheme. But if you guys are unsure, just write it down, but don't trust that thing on its own lah. Add many more points. Also, don't forget that it is sex-linked tadi, lupa tulis. It is sex-linked and it's also recessive trait. By writing down that it's a recessive trait, you guys would have gotten some marks also. Okay, anyway, next, as usual, when drawing a Punnett grid, we need to define the allele. We need to define the allele. So let's begin with the X with a small h. X superscript small h is the gene for hemophilia. The gene for hemophilia. The X with the big H is normal. Gene for normal. Gene for normal. Some note, as have been put in the mark scheme before, do not say that hemophilia is carried on the X gene. Hemophilia is not carried on the X gene, on the X gene block, on the X chromosome. Hemophilia is carried in blood. What's carried on the X chromosome is the gene for hemophilia. So be careful of that particular term right there. Anyway, as usual, let's draw the Punnett grid. So for the first parent, let's say that the first parent is, let's say that the female parent has is a normal. The first female parent is a hemophilia carrier, in the sense that they have one H, one normal H, and one with hemophilia gene. While for the male partner, it's a normal guy, your typical normal guy with one Y chromosome and one normal X chromosome. So let's look at the Punnett grid, and then let's do the crossing. So XH with a Y. So we have X small h with a Y. We have X big H and X big H. And finally, we have X big H with XH. Make sure to label, make sure to put a label that this is the female parent. Just put a label to be able to be safe, the female parent, and this is the male parent. And we know that the middle part are all the offsprings. Next, once you've gotten the offspring, make sure to first do the genotypical ratio. Genotypical ratio of the offspring, well, the ratio of offspring, the genotype of the offspring must always be expressed as a ratio. So this is to ensure that, well, it must be expressed as a ratio because that's how we do things in genetics. So to avoid us from being confused, always label the genotype. So maknanya, let's just put the genotypical ratio. Genotypical ratio. So let's look at how many genotype that we have. We have X, big H and Y. We have X, small h and Y. We have X, big H and X, big H. And we have X, H and X, small h. So from then, we come up with the ratio, and we see the ratio is 1, ratio of 1, ratio of 1, and ratio of 1. This is the genotypical ratio. So that would have given you some marks right there, but that's not enough, because the question specifically asks who is the carrier and who is the who is the people who get the hemophilia. So we need the second part. We need a phenotypical ratio. Sorry, terbahasa Melayu pula. It's PH kan? Pheno. We need to write the phenotypical ratio also. So thank God, in the cross that we did just now, all 4 offspring have different phenotypical ratio. So the phenotypical ratio that we have right now is that we have male with hemophilia, ratio with, aku follow yang kat atas so supaya dia senang nampak. So we have a normal male, and then we have male hemophilia, and then we have a normal female, and then we have carrier female. So that's a ratio of 1, 1, 1, 1. To ensure that we get the full marks for this question, we do some explanation. Maknanya, we tambah explanation for a cross between, kita tengok for a cross between female carrier, and a male, and a normal male, for a cross between a female carrier and a normal male, we would get, there would be a 25% chance of having a male. Okay, even there would be, okay, this isn't accurate lah. There would be a, sebab kita nak spesifik pada male kan, so male ada half je. So kita tengok male sebab kita ada 2 je male dekat sini. So meaning to say, among all the sons that these two parents have, there is a 50% chance that their male child, their son, would have hemophilia. So that's a very high chance actually when you look at it. And then for the same set of parents, there would be another 50% chance of having a daughter who is a carrier. So if you guys want to do the Punnett grid, make sure to explain here until here. Make sure to explain all the way from the Punnett grid to the genotypical ratio to the phenotypical ratio, all the way until the percentage chance of having these 2 traits that the question asks for. Also, if you guys still are not sure about the question, then you can always do another cross. You can always do another cross. Nampak macam lama tapi sebenarnya kejap je nak jawab ni rather than tulis essay. You can always do another cross. Kali ni kita ambil okay, I'm just gonna do the cross very quick sebab kita ada 2 period. Aku buat answer slow. Let's do another cross. Sebab in the marks, sebab should be by right, atas ni dah cukup dah 8 marks. Tapi if you guys are not sure, there's another thing that we can show. So we can do another cross. Kali ni kita ambil male yang ada hemophilia. So a male that has hemophilia, this time with a normal female. This time with a normal female. X with big H, X with big H. I'm gonna skip the label for now. Big H, so XHY, XHY, XH besar, XH kecil, XH besar, XH kecil. Alright, so if we do the genotypical ratio, we'll get XH, there's XH besar Y, and then the ratio with XH besar XH kecil, and that is a ratio of 2:2, so that's ratio of 1:1. So when we look at the phenotypical ratio, there is male, male normal crossed with female carrier, crossed with a female carrier with another ratio of 1:1. So what does this mean? This means that a carrier, carrier pula, an affected male parent will never pass on the hemophilia gene to their son. Sebab nampak, the female is normal, the female parent is normal, tapi the male yang affected. Tapi because the gene is located on the X chromosome, so a male father, a hemophiliac father, will never pass on the hemophilia gene to their son, but it can be passed to their daughter. But if the mother is normal, the daughter will be only a carrier. So there is a 100, so in this particular set of parents, there is a 100% chance of having a normal male, a normal son, but there will also be 100% chance of getting a female, a carrier daughter. Carrier daughter. So I think that's all for this question. Is that okay?
Faham.
All right, I hope that's clear. Thank you. Allow me to stop the share screen. Okay, if that's okay, we're gonna move on to the next set of question. All right, ini question siapa? Aku tak ingat. All right, so for now, I'm gonna continue for this one. All right, so again, look at the question as a whole. So again, this question is very straightforward. Describe the Down syndrome. Describe how the human skin is determined genetically, and explain the causes of sickle cell anemia. So when we look at the word describe the causes of Down syndrome and stuffs, again, describe is almost like explain. You have to show how it's there detail by detail. But how deep do we have to describe the causes of Down syndrome? Let's look at the answer scheme. So first, Down syndrome, look, it says describe the causes of Down syndrome. So there will be no marks for you to give the definition of how Down syndrome looks like. But there would be no problem if you want to do it. But anyway, Down syndrome is caused by non-disjunction. So I would like to ask Alia, what does it mean by non-disjunction? Walaupun jawapan ada kat situ kan, saya nak tanya. Hello, senyap je.
Alia pergi toilet jap.
Okay, all right. So never mind. So Down syndrome is caused by non-disjunction. So macam yang disebut kat bawah tu in the third line. Non-disjunction occurs when chromosome fails to separate in meiosis 1 ataupun the chromatids fail to separate in meiosis 2 or anaphase 2. Why do they fail to separate? This can be caused by a number of reasons, including number 1, the centromeres fail to divide, or the second one, there is a failure in the formation of apa nama benda tu? Yang tali-tali tu? I forgot the name.
Fibers.
Okay, fibers. Tubule fibers. This kind of stuff. I want to say gentian gelendong. My brain just said gentian gelendong. The Malay brain. Right. That is what meant by non-disjunction. So it occurs during meiosis. So that itself is worth 3 marks. So that's a good way to gain a maximum of 5. Next, some gametes have an extra chromosome. It has to be gametes because if you can say some cells, that's not enough for fertilization to happen. There has to be a gamete with an extra chromosome. Next, when a gamete can have an extra chromosome and then fertilization happens, creating zygotes or individual with 47 chromosomes. So istilah specific for Down syndrome would be trisomy 21, or if you can't remember the term trisomy 21, you can say that there is an extra chromosome number 21. There is a third chromosome at the chromosome number 21. So that's the thing. In IB, we'll only be covering Down syndrome, so that's cool. Just remember trisomy 21. No need to care about other syndromes. But the thing is, it will have, so this, some people probably won't remember this last point, but because it's described, that's why you can say it too. People with the probability of having Down syndrome increases with age of mother, with the age of mother, with the age of parents. Meaning to say that as parents get older, there is an increased probability of Down syndrome. Why? The question didn't ask for why, but if you want to know, it's because of genetic mutation that occurs that might cause some failure in the formation of microtubules. Okay. Any problem for question number A?
No.
All right. So question number 2, again, describe how the human skin color is determined genetically. Before we go to the answer, skin is right. So the human skin color, is it determined by one gene or is it determined by many genes?
Many.
Yes, it is determined by many genes. Examples of many gene inheritance are the one polygenic inheritance include the human skin color, the color of the corn kernel, human height, and whatnot. And usually, polygenic inheritance, if we were to take the data of the whole population, it will form a normal distribution curve. So just take note of that. Anyway, skin color. So skin color is an example of polygenic inheritance in which that more than one gene is responsible to contribute to a person's skin color. So just by that, you're gonna get 2 marks. So why exactly? We will talk about the amount of melanin in the skin. As we add, as more and more gene for skin color is present in a human body, it will increase the amount of melanin in the skin, which will lead to darker skin color. So much, which will lead to darker skin color. Anyway, this have occurred due to combination of alleles determines the phenotype. That's why I said as you add more genes, the skin get darker. So the word that we use is combination of allele that determines the phenotype. So kita dah cerita pasal cause, the first part. The second part, kita cerita pasal effect. What will we see? So they will solve for allows for range of skin color. Tapi you guys realize that a lot of us akan ingat the first part. Jadi kita akan guna the second option, continuous variation of skin color. It's not discrete variation, but it's continuous. That's why kita akan nampak dia dalam term of a normal distribution curve. And the phenotype, they do not follow simple Mendelian ratios of dominance and recessiveness because there's many genes involved. So you can say that it is a non-Mendelian ratio. So another thing about skin color is that the environment also affects gene expression of skin color. Sunlight UV can stimulate melanin production. This is an additional point. If you guys can't remember this, it's okay, but it's good to have it in your explanation because it will relate to the expression of genes later. And the more recessive alleles there are, the lighter the skin color is. Knowledge that you guys have. Lighter skin color is actually a recessive allele. Right? Any question regarding question B?
Not at the moment.
At the moment, all right. So finally, the 8th question mark, again explain. So this has to be in detail. You might be asked to draw a Punnett grid. Explain the causes of sickle cell anemia. So sickle cell anemia is it? So sickle cell anemia, we know that number 1, it's a recessive trait. And number 2, sickle cell anemia is also a codominant allele. Codominant allele that it's not present on that. It's a codominant allele. So when there is a codominant allele, bila ada satu je recessive gene, dia akan ada some effect but not the full effect of the disease. Tapi bila ada both recessive gene, baru the full range of the symptoms of the disease will be shown. Right, so let's look at sickle cell anemia. Okay, sickle cell anemia, because it is a case study, so kita kena tahu in detail which gene is actually which gene is actually responsible for the sickle cell anemia. So number one, it's caused by gene mutation. Obviously, like any genetic disease, it's caused by a gene mutation, and directly to a base substitution. It is okay if you can't remember the base substitution as a term because we don't cover it in great detail. But the most important thing is that it changes the code on the DNA, which leads to a change in the transcription, change in mRNA. So usually kita akan ingatlah the change in the protein that is produced. So it occurs on the 6 amino acid. Where glutamine, where the amino acid glutamine is replaced with valine. Just remember that the amino acid glutamine is replaced with valine. I'm not sure if you have to remember the exact code or not. I mean, if you want to be safe, just remember the exact code. But if I'm not mistaken, teacher kata usually the code akan diberi. But if you want to be safe, just remember that lah. But glutamine will be changed to valine. So itulah kita cerita from the code, which in turn leads to a change in translation, change in the polypeptide chain and protein. So the tRNA adds the wrong amino acid to the polypeptide chain, and finally glutamic acid replaced by valine. So if you notice, daripada first point sampai the first half ni, semua kita follow je sebab dia explain. So kita kena follow daripada awal, daripada transcription, sampai ke translation. Where did the process go wrong one by one? Baru kita dapat full mark. Kalau kita start dekat glutamic acid replaced by valine, nanti we'll lose a large number of the marks. So that's a problem. So when the question says explain, memang explain daripada nenek moyang sampai ke anak cucu nombor 7. Next, when glutamic acid is replaced by valine, it will produce abnormal hemoglobin. Tengok, abnormal hemoglobin pun dah dapat markah. And then explain detail lagi. Abnormal hemoglobin, shape dia macam mana? It will be, as the name suggests, sickle cell punya shape. It looks like a sickle. And when it looks like a sickle, it lowers the ability to transport oxygen. And ada banyak lagi lah sebenarnya. I'm not sure why is it not expected to be explained here. Tapi that's the main point lah. The sickle shape. The most important is the sickle shape. The most important symptom of sickle cell anemia is the shape of the erythrocyte. And as usual, the sickle cell allele is codominant. It's recessive. It's codominant. So homozygous HB and HS, they have sickle cell anemia. So when you have HBS and HBS, maknanya you have both sickle cell anemia gene, so it's lethal, meaning to say that you're gonna die or something, you're affected by hemophilia. But if you're heterozygous, you're gonna have an advantage in which that sickle cell anemia carrier are resistant to malaria. So you have to explain until that for 8 marks. Any question? All right. Okay, next. Ni siapa punya pula?
Okay. So question dia, A, B, C. A, draw a labeled diagram of the molecular structure of DNA comprising of 4 nucleotides. B, outline the therapeutic use of stem cells. C, giving one specific example, discuss genetic modification in organisms, including the potential benefits and possible harmful effects. So A dengan B dia bukan topik yang kita study, tapi tak apa, so kita revise dia balik. So untuk A, dia kata draw a labeled diagram of the molecular structure of DNA comprising of 4 nucleotides. So tengok margin dia. Next. Okay, so untuk lukis molecular structure of DNA, so kita kena make sure ada phosphate group pentose sugar, ada nitrogenous bases, ada 4. Lepas tu kena make sure ada hydrogen bond yang sambungkan nitrogenous bases ni. Lepas tu covalent bond yang sambungkan phosphate group dengan pentose sugar. Lepas tu make sure everything is labelled, so dapat full mark. So yang ni senang je. So next, untuk soalan B, tengok balik soalan B. So soalan B tadi dikata outline the therapeutic use of stem cells. So yang ni topik 1 pasal therapeutic use of stem cells. So kalau tengok marksheet, so dia katakan outline. So kita kena cerita pasal proses dia lah. Macam mana stem cells ni kita boleh guna. Macam apa fungsi dia kan. So bila tengok marksheet dia start dengan example of stem cells. Macam mana dia obtain. For example, stem cells are obtained from bone marrow or cord blood or inner side. Ini mana kalau tulis akan dapat 1 point. So why is kita explain pula kenapa stem cells ni boleh digunakan. Kata sebab stem cells they have the capacity to divide and also they are unspecialized and differentiated. So because of this ability, they have the ability to differentiate or specialize. So because they are multipotent. So okay. So lepas tu next kita explain pula disease yang guna stem cells as therapy. Macam yang kita belajar leukemia. So example of therapeutic use for leukemia, the patient's bone marrow cells are killed and replaced with the stem cells. So bila kita tulis semua ni, so dapatlah 5 marks. Okay, last question C. Giving one specific example, discuss genetic modification in organisms, including the potential benefits and possible harmful effects. So dia suruh discuss genetic modification pasal GMO. Lepas tu dia kata kena include potential benefits and possible harmful effects. Lepas tu kena kasih one specific example. So kalau nak senang tengok marksheet dia. Okay. Nak senang kita start dulu dengan definition of GMO. So kita tulislah genetically modified organisms are organisms where characteristics are altered or changed by addition or removal of a gene. So it is a reference to the specific gene transferred to the host organism. So after kita define GMO, kita bagi tahu pula dia punya example untuk GMO tu lah. So yang kita belajar is BT corn. So kita boleh explain lagi kenapa boleh ada GMO ni kan, sebab dia because of universal genetic code which allows for the genes to be transferred between species. So the gene transfer which involves splicing genes into a suitable vector or host DNA. So after placed in the host cell cycle, ni basically is the process for untuk buat GMO ni lah. Okay, dah tulis tu, kita boleh explain pula potential benefits and harmful effects. So untuk selamat, potential benefits ni kita kasih 3, dengan harmful effects pun kasih 3. So macam kalau korang dah explain tadi untuk GMO, kasih definition dengan example, so insyaAllah dapatlah 2 markah. Okay, so untuk potential benefit yang first, make sure kalau saya nak selamat, make sure the potential benefits and harmful effects yang kita tulis kat sini ada kaitan dengan example GMO yang kasih yang BT corn tu. So kat sini ditulis, first potential benefit is increase in yield or productivity. Second is allows for the introduction of a characteristic that wasn't present within the gene pool. And third potential benefit, less use of chemical pesticides. Yang ni ada kaitan dengan BT corn sebab BT corn yang GMO ni dia lagi tahan kepada pesticide, tak silap lah. So potential benefit dia, less use of chemical pesticide. So dah habis explain the benefits, kita explain pula dia harmful effects. So first is there is possibility of cross pollination. Second harmful effect, apa ni? It could have currently unknown harmful effects, or the toxin may cause allergic reactions. And third harmful effect, lastly, it could reduces genetic variation or biodiversity. So tulis semua ni dapatlah 8 markah. Okay, dah tu je. Semua faham ke? Faham?
Faham.
Alright, okay. Moving on to the next slide. Okay, this time for real, my part. Sorry, aku dengar suara aku je hari ni, first period. Okay, anyway. So, tadi kita dah tengok question yang direct, except for question last Wafi tadi, itu question tu tak berapa direct. Next, kita ambil question yang tak direct. So, first, distinguish between autosomes and sex chromosomes in humans. So, bila kita kata distinguish, kita nak differences. So, distinguish, just give the differences between autosomes and sex chromosomes. So, the first yang you guys kena buat ialah dah nama distinguish, bagi definition dulu. What's the definition for autosomes and what's the definition for sex chromosomes in humans? Anyway, oops, alright. So, terus beritahu the uses lah. X and Y chromosome determine sex. So, females ada XX and males ada XY. X chromosome is larger and carries more genes than the Y chromosome. So, the first 3 part itu kita explain just what apa benda lah XY chromosome ni sebenarnya. Finally, for autosomes, there's 22 pairs of autosomes, and males and females have the same type of autosome. So that's how you distinguish between the two. Asalnya kita cerita X and Y chromosome male and female berbeza, autosomes male and female sama. So I don't think there's any problem with that. Okay. Anyway, next question. Again, ni describe the inheritance of hemophilia, including an example. Allah kena lukis lagi. Describe an example of the inheritance of hemophilia, including an example using a Punnett grid. Yang ni specifically dia minta using a Punnett grid. Macam tadi kita ada choice kan. Macam kali ni dia minta spesifik kena guna Punnett grid. So we have to comply with that. So I think I've shown enough of Punnett grid for hemophilia. So I'm just going to show the answer scheme lah. Cuma bezanya this one, it's more general I guess. But tadi dia minta specific how males can akan boleh contract hemophilia and how female will only boleh jadi carrier. Ini kita lebih general, so we can give both. So we know that hemophilia macam biasa is sex linked sebab gene on the X chromosome. It's more common in males. Ini tambahan point tadi. It's more common in males who only receive one X chromosome. So it's more common. So female is hemophilic kalau it's homozygous recessive. And usually it's fatal lah kalau homozygous recessive. So XH for dominant normal allele, XH for hemophilia allele. So this can be shown just in the Punnett grid. Just show the Punnett grid. You're gonna save so much time rather than writing a whole essay. Just show the Punnett grid. And then half the males are hemophilic and half of the females are carrier, OWTT. I don't think there will be any problem with that. Do not allow marking point D for the letter. If the letters dominant are not upper and lowercase version of the same letter. We can use any letter to be honest for upper hemophilia, but just use H to avoid us from getting confused. And finally, question C. Explain how meiosis results in an effectively infinite genetic variety of gametes. Okay, soalan ni agak berbeza. This should explain meiosis, yes betul. Tapi it's not just explain meiosis in detail. Kalau kita explain how meiosis work A sampai Z, nanti markah dapat seciput je. Kita nak dia explain macam mana from meiosis itu kita boleh hasilkan infinite genetic variety of gametes. So kita kena tengok dalam proses meiosis yang panjang-panjang tu, which one yang akan produce genetic variety. So before looking at the answer scheme, dah boleh teka dah. Kita boleh tengok pada independent assortment of chromosome. Kita boleh tengok kepada the crossing over, pada crossing over. So kita tahu jin mana yang akan cross over. Dan kadang-kadang dia tak cross over. Among other stuff lah. And let's look at it. So first, kita cakap pasal homologous chromosome. One homologous chromosome is from the mother and one is from the father. So that's the first genetic variety. Sebab these two homologous chromosomes will almost definitely be different. Number two, homologous chromosomes are in pair in prophase 1. So waktu in pair in prophase 2 apa benda? Berlaku crossing over where there is chiasma formation. So crossing over is the second source of variation. So that itself can be 2 marks. And then apa jadi bila ada crossing over? Kita akan ada recombination of linked genes. Yang mana bila homologous chromosome between the father and the mother is crossed over, there will be a transfer of genetic material, creating the combination of linked genes. And then ini yang is not covered as much in our syllabus, but please remember this. There is many possible points for crossing over. Sebab crossing over bukannya dia cucuk hujung ke hujung je, but crossing over can occur at any point in the allele, in the chromosome, because we know that one chromosome can carry dozens and sometimes hundreds of genes. So any one of these genes could be a possible point for crossover. So itu dah banyak dah variation here. And the best thing is crossing over occurs at random position. Mana-mana je dia akan pilih, tak kisah dekat mana. So even more possible variation. So due to crossing over, the two chromosomes of metaphase 1 chromosomes are not identical. So bila dah berlaku crossing over, dua-dua chromatic tu dia dah tak identical. So nanti bila dia split, there will be even more genetic variation. And then next, waktu metaphase 1, metaphase 1 ni dia beratur kat tengah kan. So there will be random orientation of bivalence in metaphase 1. Meaning to say that chromosome from father ke mother ke, ikut dia lah, nak duduk kiri ke, nak duduk kanan ke, nanti bila dia pecah, dia dapat yang berbeza. So the orientation is different. So since the humans have 23 pairs of chromosome, that would be a lot of difference for each gamete. So waktu anaphase, at the end of metaphase 1, chromosome move to the opposite poles, and before they move to kena independent assortment of chromosome and genes. Jadi same explanation for tadi lah, dia berlaku independent assortment of chromosome. Maknanya dia akan berkumpul semuanya berbeza than one another. So this would create how many? 2 or n ataupun 2 to the power of 23 combination without considering crossover. Maknanya kalau crossover tak berlaku pun, kita akan ada 2 to the power of 23 combination. That's large enough. With crossing over, jauh lagi banyak. And we will have 4 genetically different nuclei gamete from each meiosis. And if each meiosis berlaku lagi banyak. So that's how you explain how from meiosis will have an infinitely different combination for gametes. So enough sampai gametes saja. Kalau diminta sampai offspring, you guys boleh explain all the way sampai fertilization, dan the fertilization, the number of sperms and whatnot. Tapi disebabkan dia minta gametes, enough sampai gametes sahaja. Oh, and ini fun fact sikit lah. Except any of the points above in a clearly annotated diagram. So if you guys rajin nak lukis meiosis one by one and annotate it, that's also a good way to do it. Any question? Any question? Laju sangat ke?
Okay je.
Okay je. Alright. Okay. Last question of genetics before kita habis. Last question for genetics. Again, ni soalan yang lagi tak jelas. Kita back to basic, kita sembang pasal Mendel. The biological insights of Mendel and Darwin in the 19th century remain important to this day. So question 1, dia suruh discuss kena ada, usually kita kena ada good and bad lah. Kita tengok apa yang dia nak discuss. Number 1, the role of genes and chromosome, but role of them. Kita tahu role of gene and chromosome ada banyak. But in what? In determining individual and shared character features of the members of a species. So ini dia akan relate balik dengan topik evolution, speciation and stuff. So it's not specifically human, so kita nak discuss the role of gene and chromosome dalam determining individual and shared character features of members of a species. So kenapa members of a species ada shared character? So kita tengok each one, gene dan juga chromosome. So gene, mutation, changes gene which can cause genetic difference. Kenapa ada difference among the same species? Sebab ada mutation yang berlaku. And this can create genetic difference. And genes can have more than one allele. So if you guys nak explain that better, boleh juga bagi tahu definition of allele. Alleles are different forms or versions of a gene. And different alleles of a gene give different character. And that creates variation among individuals. Kita pasal allele and how they change individual characteristic in the same species. So boleh bagi contoh. One example can give you one mark. So boleh bagi eye color or any allele that you want. Eye color ke, telinga ke, skin color ke. And alleles may also be dominant or recessive. So that's one way of explaining the genes lah. See, ada banyak point. Ni berapa markah ni? 7 marks. So anything can happen. Next, point number F. Both allele influence the characteristic. Kalau codominant. Tadi kita cerita allele biasa, dominant recessive. Tadi kita cerita allele dominant recessive. Second one, LRC, okay. The second one, kita cerita allele kalau di codominant pula, kita akan both allele akan influence codominance. Dan kalau you guys sebut je polygenic inheritance pun dah okay. Nampak tak? Soalan ni you guys tak explain in detail. Kita bagi macam point form je. Ini function number 1, function number 2, function number 3. So kita discuss je apa role, apa function of it. So next, kita cerita. All members of a species are genetically similar. They have shared genes. Genes mereka sama. Tapi the expression of genes yang berbeza. Point number H. So kenapa benda tu boleh berbeza? So kita cerita, just kita buat je any reference to epigenetics, methylation, acetylation. Not all genes are expressed in an individual. So tak perlu explain in detail macam mana methylation, macam mana acetylation. Mention sahaja and you'll get 1 mark. Kalau explain in detail, masih 1 mark. And finally, genes are inherited from parents, passed on to offspring from generation to generation. Itu baru role of genes. Tapi dia nak 2 benda, genes dan juga chromosome. So kena explain juga the role of chromosome. Memang jenuh sikit lah soalan macam ni. Sebab tu kalau boleh, jangan pilih soalan yang tak clear. Tapi boleh. So chromosome pula apa? It's apa, kena cerita chromosome, definition of chromosome. Ialah same loci, same position of genes. Dekat dalam chromosome tu, kena ada same locus of position of genes. So we have same number of chromosome in a species. So, contohnya human and 46. Dia cerita pasal chromosome tu apa. And for some reason dia accept juga yang some individual ada extra chromosome and that leads to Down syndrome. And kita boleh cerita pasal anomalies yang polyploidy. You remember polyploidy yang mana the chromosome number of a species can randomly double or triple. It can create a new species. And then the X and Y chromosome determine the sex and gender of an individual. Same species, tapi gender berbeza. So itu pun variation lah. And meiosis, independent assortment, fertilization, all of this thing can give new combination of chromosome or genes. Right. Soalan ni sangat panjang. Tapi if you guys can write many of this down, you guys should gain that 7 marks. Any question for question A? Semua hidup lagi ke tu?
No question lagi.
Alright, good. Okay, sorry emang panjang sikit Right, next. Kita revise topik speciation pula. Outline the process of speciation. Kita ceritalah how species is created. So B. But first definition. Speciation is the splitting of a species into 2 species. Satu species, tiba-tiba lepas many years, kita akan split into 2 species. So next. Reproduct. So macam mana boleh berlaku perbezaan between the species? because of something called reproductive isolation. Maknanya dua species split between something, so they cannot interbreed. So the isolation berlaku kenapa? Maybe due to geography, maybe due to behavioral, tapi ini dibagi usually geography lah. So there can changes in reproductive behavior, reproductive timing. So these two group do not interbreed with one another. Lepas ni 5 minute break boleh tak? Boleh. Boleh. Let's go. Alright, next apa polyploidy. Another way, macam tadi reproductive isolation secara normal. Kadang-kadang isolation juga berlaku sebab polyploidy. Tiba-tiba katak tu dia doublekan number of chromosome dia. So they can no longer interbreed with one another. So polyploidy can also cause isolation. Next, because of that, kita akan ada the separation of gene pools. Gene A berbeza, gene group A berbeza, gene group B berbeza. So gene pool berbeza, and that will lead to differences. So gene pools that will lead to differences between the two species. So boleh juga kita cerita pasal differences, how disruptive selection cause traits to change and diverge. And then finally, gradualism. As time goes on, setiap satu species tu undergo small mutation. So as time goes on, mutation gradually accumulate sampai semuanya berbeza. And finally, punctuate juga ada dia ada dua cara. Satu gradualism, satu lagi teori punctuated equilibrium. Ada satu spesies tu tiba-tiba jadi lain. Out of nowhere tiba-tiba jadi lain. So itu punctuated equilibrium. Benda ni 4 marks ya, tapi kena cerita setiap satu proses yang berlaku. Semua benda yang kita belajar. Untuk soalan outline ni ibaratnya korang tulis setiap satu understanding from each topic. That's how you answer the question outline. Bukannya you explain in detail. Right. So 5 minute break before masuk C ke? Alright, let's go. Okay, abaikan. Aku nak flex gambar anak saudara aku. Ni gambar Aura. Dia tengah lompat-lompat atas paddle. Nak flex gambar anak saudara sendiri. Ni gambar Aura mandi kolam. Ni gambar Aura mandi kolam. Ni gambar Aura lagi. Main dekat dalam. Main dalam kolam. Nama penuh dia Aura Amalia binti Muhammad Faiz. Nama ni Aura Amalia binti Muhammad Faiz. Yang binti Arisma tak ada dah. Dah. Lagi. Nah, gambar orang ada kambing.
Ni gambar sendiri pun tak. Obviously I have a very small.
So, tengok ada orang happy tengok kambing. Come on. Come on guys, bila kita nak happy macam dia.
You know what it means.
Oh yeah.
Yes.
Dia nak ART.
Dalam satu kelas.
Kambing. Saya juga tak tahu ya.
Kalau kita ada kerja pun
And it's my turn.
Lompat. Okay dah, enough of Aura. Bila nak bahagia macam Aura. Alright. So,
aku macam 180 je.
alright. Anyway, thank you for that selingan of gambar Aura. Harap teacher tengok live stream. Alright, anyway. Kita masuk balik ke question C. Right, Bismillahirrahmanirrahim. Right, last question for the day. Before we kahoot and yeet ourselves and our souls to the devil, describe using one example how homologous structure provide evidence for evolution. So again, soalan describe, tapi there's a catch. Wajib bagi example how homologous structure provide evidence for evolution. So if you remember, homologous structure are structures that is similar across different species. Contoh paling famous ialah, tengok kat tangan sendiri, pentadactyl limbs that is present across many species. Ada kat humans, ada dekat whales, ada dekat burung. Pentadactyl limbs. So that would be an easy example that we can look for that can provide evidence for evolution. 4 marks je, tak payah fikir banyak-banyak. Oh shit, sorry. So question C. So A, similar, so bagi definition dulu apa-apa. Similar structure but different function in homologous structure. So tengok macam dia kata, nak senang, sebut je pentadactyl. Ingat satu example and your life is complete. Pentadactyl limb. Kalau tak ingat sangat nama pentadactyl tu, ingatlah 5 jari. Limbs with 5 digits. Digits ni bukan digit MAA. Digits ialah refer to, siapa yang belajar French tahulah perkataan dua. Dua means jari ataupun apa, jari lah senang cerita. Digits or toes. So apa yang ada dalam pentadactyl limb ni, they have similar bone structure. Kita bercerita pasal example of similarity but different uses of function. So ceritalah dekat whales, the pentadactyl limb tu jadi dia punya flipper. Dia punya apa? Dia punya flipper, dia punya fins. In humans, the pentadactyl limb is used for locomotion dekat tangan. In birds, the pentadactyl limb is used to terbang. So pentadactyl limb. So give 2 examples. Terdapatkan 1 mark of pentadactyl limb by a vertebrate group. So whales and humans cukup di situ sahaja. So ingat, tak boleh cerita saja. So dia nak evidence for evolution. So maknanya kenapa different species ada sama juga. So this suggests that these two has a common ancestor And yang paling penting ialah wujudnya evolutionary divergence, in the sense that there is a common ancestor and then speciation occur and evolutionary divergence happen, split macam tu. So this is what it suggests. So this process is called adaptive radiation. So macam mana nak ingat benda ni? Adaptive radiation. Kita dah tahu evolutionary divergence, benda tu berpisah. So radiation, it will give you an image of something yang kat tengah macam matahari, benda tu radiate keluar. So that's why it's adaptive radiation. Benda tu apa, one species radiate, berpisah kepada banyak species to adapt to the changes in the environment. So I think that's all for genetics that we will do. Any question?
Tak ada, faham dah.
Tak ada? Tak ada. Alright, thank you. Sorry kalau tak clear. Okay, dah. Saya pernah buat Kahoot. So, nak buat Kahoot tak? Wafi Wafi, ada Kahoot tak? Nak untuk dibuat? So, tak jadilah ni nak habiskan awal ke? Ke next topic? Next topic korang nak buat apa? Kita ada a few more classes. Kalau tak boleh kita allocate this time untuk cari soalan for the next topic.
Well, hari tu second.
Bola hari tu second apa yang lain?
Kejap, saya check. Tak, saya ada screenshot lah, I think, and send sana. So, siapa jumpa duluan? Cakap lah. Gitu. Okay, ni. First genetics, second plant biology.
Okay, so kita buat soalan plant biology. Lepas tu division of part bulat-bulat, part factory recall, part data and part essay kita buat dalam group je. Tapi kita tukar-tukar lah kali ni. Jangan buat essay je. Rotate Let's go. So if that's okay, kita boleh aku assume kita dedicate baki masa yang ada ni untuk cari soalan. Let's go. Alright.